你好，很高兴见到你。Is pronunciation is right? Okay. My daughter is learning the Chinese, and she taught me this sentence before coming here. Okay. I'm Dr. Park from South Korea, Kaeming University. I have 3 topics about the female infertility and pregnancy outcomes. The first topic is first pregnancies with a simplified IVF process, a crucial step to universal and accessible infertility care from Belgium. The authors claim that infertility is probably the most neglected health care issues in the developing countries. In the developing countries, ART are sometimes unavailable or sometimes too cost. So they want to develop a cheap and simple process. They named the TV lab system. They prepared 2 vacuum container tubes. One tube contains a culture medium, and the other contains citric acid and sodium bicarbonate to generate carbon dioxide. They connect them to tubes and placed on the heat block. After oocyte aspiration, the semen were inseminated using the syringes. They can monitor the embryonic development under the microscope without opening the cap, so they can maintain the stable condition. After developing this system, they compared the result with the routine IVF culture. The primary outcome parameter was day 3 embryo, and second outcome parameters were embryo implantation rate and ongoing pregnancy rate. They enrolled only first IVF attempts in women under 36 years old with more than 8 oocytes, and they performed only single embryo transfer. Here's the result. The fertilization rate and cleavage rate are similar in both groups. In TV lab system, top quality embryo were produced 66%, implantation rate is 35%, ongoing pregnancy rate is 30%. And then they also reported the first livers with using this system. Here's a full comparison. They enrolled 35 couples. Some oocytes are cultured in TV lab system. The others are cultured in routine culture. The fertilization rate and implantation rate is quite comparable. So here's conclusion. IVF methodology can be significantly simplified and result in successful outcome comparable to the routine IVF. The cost of TV lab system is just €200. It's just 10% of the Western style IVF program. I found another articles about the simple culture. It comes from Japan, and it was published on last year. They put the culture dish with the deoxidizing agent, and then when the oxygen concentration is below 5%, then seal up the plastic bag and placed on the warm plate. The result is amazing. They produced 99% of 2-cell embryos and 93% of the blastocysts, even though they didn't control the carbon dioxide concentration or humidity. We've been heard about lots of the timeless system today. It's a completely opposite side of the movie. Well, anyway, who knows, we can curse it in a simple way in the future. Okay, I move to a second topic. The second one is the genotyping analysis of polymorphism of the annexin A5 gene in patients with recurrent pregnancy loss from Japan. Previous cross-sectional studies have shown that recurrent pregnancy loss is associated with polymorphism at the annexin A5 gene. The authors want to know whether the risk alleles influence on further risk of miscarriage. They performed the genotypic analysis in 192 recurrent pregnancy loss patients and 195 fertile controls for 6 SMP in the annexin A5 gene. Then they compared the subsequent live birth rate between the patient with and without the risk alleles. Genomic DNA was extracted from the peripheral blood, and logistic analysis were performed to examine the association subsequent miscarriage with the variable factors and the SMP. Here's the result. They found SMP5 is significantly higher in the recurrent pregnancy loss patient. The live birth rate of patients with and without the risk allele of SMP5 were 77% versus 81%. So they conclude that adequate selection of patients based on the analysis of several kinds of SMP might improve therapeutic effectiveness. The annexin A5 promoter, especially SMP5, is significantly related with pregnancy loss. So they should be included in the analytical panel of inherited hypercoagulation factors. Thrombophilia in recurrent pregnancy loss is emerging as a hot issue nowadays. However, there is no consensus about what kinds of genes we can study, and also there are big differences between the ethnic group. I found another articles about the polymorphism from China. It was published in this year. They investigated the polymorphism of antithrombins, thrombomodulin, tissue factor pathway inhibitor, factor 5, factor 2, and annexin A5. But they failed to find the increased risk of RPL in annexin A5 polymorphism. So we have to study more about this. My third topic is human embryo release extracellular vesicles, which may act as indicators of embryo quality from UK. Nowadays, we are moving to a single embryo transfer, so we need more objective, non-invasive method of embryo selection. Cells release extracellular vesicles. Extracellular vesicles have a role in cell-to-cell communication and are involved in many physiological and pathological processes. So they have potentials as a biomarker. Nanoparticle tracking analysis provide real-time visualization and characterization of EB size and concentration. The authors tried to demonstrate the presence of extracellular vesicles in embryo culture supernatant and compared and investigated the relationship of EB size and concentration with the day of embryo development and morphological quality. They detected DNA using the intercalating dye and characterized the EB size and concentration by NTA. Routine clinical morphological grading was performed on 239 embryos from 18 couples. Samples of the embryo culture supernatant were collected and analyzed by NTA. Here's the result. The embryo supernatant contained membrane-derived EB with more 85%. The median size of EB is significantly related with the embryonic development. Increasing median size of EB was strongly associated with poor quality. So slow developing embryos on day 3 show a significantly increased in EB size. Their EB measurements were similar to embryos with good quality, and they found the presence of DNA in the EB. So they conclude this finding might lead to complementary, non-invasive method to assess embryo quality. EB characterization may predict recovery of slow developing embryo and oocyte quality. Even though they didn't check up the implantation rate, they presumed the EB may be a key way in which the embryo signals to the endometrium. In that case, these results may have important implication for both IVF protocols and determine the implantation potential of an embryo. There are several types of extravesicles, such as exosomes, microparticles, apoptotic bodies, and so on. Extracellular vesicles contain surface protein-like receptors, ligands, and also contain the DNA, microRNA, what we heard about in the early session. So considering the EB size in this study is not an exosome, so I think they need to check up the surface protein to what it is, define what it is. Thank you for your attention.
Okay, we thank Professor Kim a very briefing report for this session of the update female infertility and pregnancy outcomes. I think these three episodes was very interesting and covered from the cultural system to the morphology of embryo, right? So is there a question or comment from the floor? Yes?
I'm the Xiyuan Chen from Taiwan. I would like to ask about your lab culture system, because I see the data, the new system, simplified system, seems to have higher implantation rate compared to the conventional lab. Would you like to talk about more how the system works, how to do the system?
Actually, I have also asked about the question to the authors, but they didn't explain it well. I'm sorry.
Did they publish?
No, not yet.
Not yet?
Yeah, just
Okay.
presented in the oral sessions.
But we can recognize a lot of effort try to make the reproductive medicine more friendly, more easier. Not only about the regimen, the stimulation, the culture system, and the methodology. Yeah. So is there any comment or question from the floor? Okay, Professor Chen again.
And you have talked about the extracellular vesicle, maybe a signal to enhance the implantation. But your presentation says if the embryo maybe secrete the medium size, EV, is a bad embryo. How do you correlate these two meaning? Because it's bad, it's all good. The extracellular vesicle has any physiological significance in implantation?
Actually, we are focused, it's not my study, actually. So
Yes.
nowadays, we are focused on the microRNA or the extracellular vesicles. Extracellular vesicles contains of surface markers and receptors, ligands, and also microRNAs. So it is very important to have a role in cell-to-cell communication. So what I mentioned that they need to characterize what it is. If it is exosomes, as you say, it's have regulatory functions to inhibit the immune modulation or gene expression or like that way. But the authors declared that it contains of DNA, so it's not an exosome. So it could be microparticle or apoptotic body. So we don't know that. If it is an apoptotic body, then the EB size is increased, means full quality. If it is microparticles, then we can explain it what it is. So my comments is that they need to characterize, define what it is. Okay.
Great. Okay, any further comment? If none, so we thank the Professor Park again, for his great contribution for this.